Mr. Vamsi. Morally.
Carry on. Continue. You are audible. Continue.
Size
Arna, continue. You are audible. Please continue.
Acha. Let's go first. A very good morning to all. I cordially welcome all the honorable members, distinguished speakers, and all the participants from different countries across the globe for the second day international webinar. Firstly, I would like to thank my nephew, Mr. Deepak Vamsi Peravali, Analog Layout Design Engineer, Apple Inc., Dallas, USA, for suggesting a wonderful, multi-talented, renowned, and eminent person as a resource person. Thank you, sir, for your great concern to accept our invitation in spite of your hectic schedule. Thank you so much. Now, we'll know the biodata of our today's resource person. Dr. Anna Gulio is a lead software engineer and architect with a medical degree, Invitee, Scalable Genetics and Oncology, Azerbaijan Medical University, Dallas, USA. He is a full-stack software engineer with 19 years of experience, a well-qualified architect, business analyst, team leader with medical degree and scientific background. He is familiar with coding languages and tools, namely Python, Java, JavaScript, Oracle, DigitalOcean Cloud, Docker, microservices, and many more. He has 13,338 followers. Educational qualifications. He finished a technical science course in the year 1994. He received degree with honors from Azerbaijan Medical University in the year 2000. His experience. He is founder and technical director of Terrasoft.az from August 2008 to August 2011 for about 3 years, 1 month at Azerbaijan. He is a lead application engineer in Medidata Solutions from November 2011 to July 2017 for about 5 years, 9 months in Greater New York City. As a lead software engineer in Laurel Road from July 17 to November 2018 for about 1 year, 5 months in Greater New York City. He worked as a senior software engineer in GLG. GLG means Gerson Lehrman Group from November 2018 to May 2019 for about 7 months in Greater New York City. He is a lead software engineer and architect from July 2019 onwards in Scalable Genetics and Oncology. He developed software platform for medical genetic testing and interpretation. He rendered voluntary service as scientific communicator in NJ Science Circle from October 2015 to August 2019 for about 3 years, 11 months. He has written so many books and published honors and awards. He was finalist of Fame Lab International and Beautiful Science issued by British Council in the month of March 2009. He got multiple awards at international tournaments issued by GameTV.az and etc. He participated in brainstorming and trivia competitions. He knows 5 languages like Azerbaijani, English, French, Russian, and Turkish. Thank you, sir. Thank you, one and all.
Thank you very much. Thank you very much for invitation. Is it now time when we start the presentation, or should I wait for something?
Sir, please start your presentation.
Yeah. Again, I want to thank everybody who helped this happening. And before I start, so you can hear me well?
Yes, yes,
Is it
yes.
It's clear, sir. Yeah, it's clear.
Yeah, all right. And also let me share the presentation. One moment. How do I do it? Present another screen. Okay. Are you able to see my presentation now?
No, sir, not yet.
My question, are you able, is it possible to?
No.
Sir, can you send it to my, can you send it to me? Then I will share.
Let me.
Call us mail ID. Please send it.
One moment. What about now? Is it seen now?
Okay, sir. Okay.
All right. Very good. And
Yeah.
so a bit of solving the logistics. In the last several decades, a lot happened in biological science, medical science, and computer science, which actually helps us now to, if not understand very well, but to understand better and to use our knowledge in genetics. And that's what I named Decoding Human Software, because frankly, when we develop a very sophisticated software here on our end to study genetics, the more I work with it, the more I see that we develop sophisticated software in order to understand another software system, the software system which we all know was created by our creator, by God, and put in all living organisms, humans including. This is very sophisticated. This is something which we still just scratches the surface. And we know that genes encode proteins. And that is an interesting fact which I noticed, that the probability of getting a protein which will work, which will be useful for life, is extremely low. Look, I bring here 2 quotes from a scientific book, which it is described in the lower bottom corner with the name of the book, Undeniable. And it actually refers to a different study, a study by Mr. Denton named Evolution, which was done previously in 1980s. And the study was about how many different protein molecules existed during all timeline on Earth. And this is the position of evolutionists. And the calculations show that probably we had 10 power 40 protein molecules existing on this planet if the timeline of life on Earth goes according to evolutionists, which personally I do not agree with. But 10 power 40, it's a huge number, right? And now the question, if those proteins were rearranged in some random way, how many times attempts to get beneficial protein we had? 10 power 40. Okay, that's a very big number, lots of attempts. But what is the probability of getting a useful protein, a protein which has some function and can perform that function? What is the probability of getting such protein by randomly rearranging amino acids? And the study, that is another quotation on right bottom corner, that you see another quotation. The study shows that the probability of getting this randomly is 10 power 74. So if we combine these two numbers of how probable is to get protein randomly and how many random attempts will happen during evolution, we see that there is a big gap, a gap of 10 power 32, no, 10 power 34. That is how improbable it is that it happened in a random way. That inspires me to understand that the software inside humans, inside all beings, is actually developed. What we studied, why we studied, what we did understand. Okay, so figures like humans share like 60% genetic similarity with bananas and other very far from us physiologically and anatomically organisms, they are kind of correct. Housekeeping genes are genes which maintain basic functions, functions related to dealing with water, with oxygen, with nitrogen, with different micronutrients. Actually, genes which are dealing with ecosystem on this planet are largely shared across all species. That's why we share 60% genetic similarity with organisms like many plants, even, or fungi, which are very different from us. If we come to mammals, and mammals definitely anatomically and physiologically quite similar to us already, the similarity comes to like 90%. Only like around 10% of genes are different between me and a cat, or me and a cow. And humans share more than 99% of genes. Roughly, like humans, or even of different races, they may differ from each other roughly like 0.5% of genes only. Very small amount of difference. Most of genome is very similar, and that actually helps us when we study and make the analysis of genetic material. So we were able to map the genome of humans, meaning all genes of humans, and any random person taken on this earth will have just several million places where he is different from that reference genome. That's what we name a reference genome. It is what was generated by kind of statistical analysis of several different people, but people are not that different from each other genetically, as we see, like less than 0.5% usually, which is from 5 to 25 million places which are different. And different genes, of course, have different variations. And we know currently around 90 million different variations of different genes. But of course, each person doesn't have all of those variations. So again, but when we speak about genetic similarity, it's important to emphasize that it's about genes specifically. And genes is a very small portion of DNA. Actually, the coding parts of genes, the exome, so-called, it is 1.5% roughly of our DNA. So these numbers of similarity, which we were mentioning, it's mostly about this 1.5% of our DNA. If we talk about whole DNA sequence, then no. With bananas, it's not 60% similarity. It's just slightly above 1% similarity. Non-coding DNA, it is something which is still a dark matter for genetics. We don't really know what it is for and how it works. It is considered by many scientists that it can be junk and it doesn't have work at all. but no proof to it. And most probably, like from my opinion, there is some function. We just didn't stumble upon it. Well, how big is human genome? If you heard that human genome is roughly 3.2 billion nucleotides, you know, DNA molecule consists of nucleotides. It is partially true, but like every cell in your body actually has double of it. We understand why, because we have 46 chromosomes, right? But when we talk about genome, that's 3.2 billion, we take half of them because like half of them is just duplicate of another. So we take 22 heterochromosomes plus X and Y, which are gender chromosomes. And we can also add to it mitochondrial DNA, because we have the DNA in our mitochondria also, not only chromosomes, but it's very small. So roughly it will be 3.2 billion. But every cell of our body consists twice of that. And if you write all nucleotides in that genome of human in a book, it will be a book which will be probably as thick as a 40-story building. So it's a huge amount of information. Thankfully, we don't need to describe all of it because, as I mentioned, humans do not have that many differences from each other. So we can use a reference genome, and for every person, we can tell only the points where that person is different from reference genome. So why do we need it in general? The changes are needed. The genetic analysis is needed for medical purposes, of course. First of all, we all carry many variations of genes, and many of those variations are defects. Defects which lead to some compromising function or even non-functioning of certain gene. Every person has several hundreds of such defects. Most of them are subclinical, so they do not manifest in any disease. A person lives even without knowing it, but statistically, every person has from 1 to 6 variants which do increase probability of certain disease or certain medical condition, between 1 and 6. And definitely testing genome of a patient, in cases of certain medical condition, does help, one, to make a diagnosis, and second, to adjust the treatment strategy, the strategy of how the disease is addressed and mitigated, medically speaking. So that is important. And in order to give the idea of the scale of that need, well, 300 million people worldwide have some rare diseases, out of which 3/4 almost are, like 72%, are genetic diseases. So there is a big need for that. And that need will only increase because of cultural changes. You know probably that in modern world, you notice that people tend to marry and to have children at a later age. Medically speaking, very bad, because the elder the age of parents, the more is probability of passing a certain genetic mutation to children. On average, it is considered that every generation, from one generation to another generation, passes like 175 mutations of genes. But with growth of age, that increases. That, interestingly, it was known long ago that chromosomal aberration, like Down syndrome or other chromosomal aberrations, were very directly connected to the age of mother. And it was known that if at age of 25, the probability of Down syndrome, for example, which is chromosomal aberration of 27 chromosome, so it was 1 in 1,250. If a woman conceives at age of 40, the probability becomes 1%. It becomes like 12 times more probable of passing this chromosomal aberration. Down syndrome is the most common, but of course not the only one. There's so many different chromosomal aberrations. It was thought that it is not so with genetic mutations, but it was found that, yeah, actually genetic mutations are also more probable at elder maternal age. And it is considered that most probably it is because of not with age, the function of repairing DNA damages, and DNA damages, they happen all the time in cells. So we have reparation instruments inside our cells. So this repairing process doesn't go so efficiently. That's why those mutations may stay with higher probability and be passed. For every year of age, for maternal age, 0.37 more mutations will happen statistically. For father, interestingly, because sperm cell production happens more dynamically, and it happens all the time. We know the oocytes are formed early, even during embryonal period, and they are just used during all age of woman. But because sperm cells are produced during all life, so that probability of passing genetic mutations on father's age is higher. So chromosomal aberration is not that, there is no that correlation, but for paternal age, there is 1.5 new mutation may pass probabilistically with every year of paternal age. So we see that the problem of more genetic conditions will be just worse, because one, with just every generation it becomes worse. In ancient times we had fewer of those bad mutations, and also because the changes in society very badly, for me it's very sad, leads to people marrying and having children at a later age. Now back to technology, right? So how does the DNA analysis work? How is it made? The very first what we do, well, we identify the patient needs genetic analysis based on clinical picture, on physiological biochemical analysis, etc. And after that, we get the sample. The sample is usually blood or saliva, because saliva contains many lymphocytes, leukocytes, which also have DNA. So we can take blood or saliva sample, and we can send it for genetic analysis, DNA sequencing. And after that, from sample, we do the sequencing, we get data. But after that, we should understand the data. Interpretation of data is a huge work, which actually became possible to do it on scale just recent several years. That's why we'll see later how the cost of genetic analysis dropped literally like 10 times, even more than 100 times in the last 10 years. We'll see it later. So first of all, extraction, right? So we receive the blood sample, and of course blood sample doesn't contain only DNA. There's so many lipids, proteins, carbohydrates, like salts, etc. We use different chemical processes in order to extract DNA. It's a technology which did exist for a very long time. It's not something new. And after that, well, we want a sequencer. Right? How does a perfect sequencer work? With perfect sequencer, you'll just put those DNA molecules, and it will give you all the sequence of nucleotides. And as I told, it's 6.2. Okay, you can take into account only half of it because chromosomes duplicate each other, and the difference is not that big. Okay, 3.2 billion nucleotides. It will be book 40 stories tall, that thick book. It's a lot of data. But the problem is that the sequencing process cannot digest the whole DNA molecule of human. because of problems of the technology. We can sequence only a part of DNA which is just several hundred nucleotides long. The longer it gets, the more noise during the technological process, and we cannot handle the whole DNA molecule. The process of sequencing actually was developed quite long ago, in 1977. Mr. Sanger, Frederick Sanger was an English British scientist, and he received 2 Nobel Prizes for his work in biochemistry and chemistry and biology, physiology. And the sequencing, well, I was always interested how it is done, right? Because, okay, we understand the DNA molecule. It consists of multiple nucleotides, just like any Lego construction consists of multiple Lego pieces, but it's so small, we don't see it, right? So how can we understand what is the sequence of those nucleotides in DNA molecule? And it took genius to understand what we can do to read DNA. First of all, there is a so-called enzyme DNA polymerase. DNA polymerase, what does it do? It works in our cells when the DNA needs to be replicated. When our cells divide, the DNA polymerase sits on DNA molecule, moves along it, and catches new nucleotides and connects them with each other according to complementary, so that those nucleotides will be complementary to the DNA chain on which it builds a copy. So DNA polymerase can build a copy of existing DNA molecule. First, it opens it as like making it a fork, and on one chain it builds a copy of it. Okay, so DNA polymerase, if you add the DNA molecule and DNA polymerase and a solution with nucleotides, it will build a second DNA molecule. Nice. Can we use it somehow to read, to understand the sequence? The idea with which Sanger came, here is kind of again illustration how DNA polymerase will pick those nucleotides and build new DNA molecule. Okay, so the idea with which Sanger came is to use not only regular nucleotides, but to mix them with the modified nucleotides, which are dideoxynucleotides, which has the portion which fluorescent. Let us see. Okay, these are molecules of those 4 nucleotides which are modified. You can see those colored pieces. They have the fluorescence. So the fluorescent dye is a part of that nucleotide, but on another side, this nucleotide is a dead end. It is terminated. So if that nucleotide is picked by DNA polymerase and used as a piece of synthesizing the chain will end because the next nucleotide cannot connect to it. And if you mix all this, well, that's how it will be. Yes, your DNA polymerase will start synthesizing your DNA copy of your DNA molecules. That is on the top of the DNA molecule which you study, which you want to try to read, and it will pick several correct nucleotides, or usual nucleotides, and then it will pick those modified nucleotides which are dead end, and it cannot synthesize it anymore. So if you have multiple of such copies of your studied DNA molecule, the molecule which you want to make sequencing, and you mix it, you have many different copies of different lengths, because just probabilistically, randomly, those the fluorescent, luminescence carrying nucleotides, which are dead end, can be incorporated into copy on different steps. So what you have many copies, and you see they just finished at different steps, and they do not go anymore. And these nucleotides have a fluorescent piece on it. It has a dye. So what you can do after that with those copies? First of all, they are of different size. If they're of different size, they will react differently to electrophoresis. Electrophoresis allows you to pull them through capillary, and depending on different size, they have different inertia and different possibility to move through the solution. So they will stick in your capillary under electrophoresis. They will stick on different levels depending on their size. So what you do pretty much, you sort them by size. And you see how much already on left side, if you try to read, you can understand what does this DNA molecule look like, yes? Because you have those letters on each level. So after you sort them in your capillary during the electrophoresis, you have bigger on one side, smaller on another side. You can just take a laser, and it is already pure physical technology, right? You use a laser, you use a light detector, and you can read those fluorescent pieces of nucleotides which are inside your capillary. And this reading allows you to pretty much to read the copy of your studied DNA, which you were analyzing letter by letter. So that's the whole process. That is the process of Sanger's DNA sequencing. Of course, years passed, we developed many other variations. Like Sanger's sequencing is pretty good. There are other technologies, virus sequencing, sequencing by synthesis, sequencing by ligation, ion semiconductor sequencing. They are all used in technology of DNA sequencing nowadays. All of them still cannot be used to read the whole DNA of human from beginning to the end, because the more you get those, the longer will be your piece. For example, think about Sanger sequencing. You will have more of noise in that noise in the light emitting, so you'll not be able to read it well with light detector, or even in the solution where you will use your polymerase. Again, you need a smaller piece so that this mixture solution of nucleotides will work well on synthesizing the copy of it. With longer DNA, it will not work well. So the longer piece you get, it doesn't work well. So what we started doing actually recently, relatively recently, we started to read sequence pieces of it and then use a software, computer software, to connect those pieces. We're going there. We'll see how it is done. So cost per human genome, as I told, it dropped more than 100 times. You see how in the year 2001, to make sequencing of human genome was more than $100,000 for one person. And right now, you can do it for roughly 1,000. This is faster than Moore's law, which is famous, Moore's law for computer technology, which states that every year, everything doubles. It is more than doubling. And it happened roughly in these years. You see, 2007, 2008. What happened? In the year 2008 to 2007, the company Illumina in America, they made their new generation sequencing devices, which harnesses this new technology. So first of all, sequencing is done as according to technology which we just looked, the Sanger technology, or several other modifications. Sequencing is nowadays already made in small chips, which are quite good. But again, they are good at handling small pieces of several hundred nucleotides. So after we extract DNA in our samples, we need to do what we call sharing. We split it into small pieces. And what we use for it is pretty much sound. So kind of the devices which shout, yell on that DNA with high amplitude, certain frequency, and I break them in pieces which are several hundred nucleotides long. What is after that? Well, after that, we should be able, for each piece, we should be able to handle it. And by the way, one more innovation that nowadays we don't need to make DNA sequencing of one patient at a time. We can make it in bulk, mixing them all together. You're asking how? If we mix all those DNAs of different patients, of many patients together, it will be a mess. How do we separate them and how do we understand? We use barcodes for that. Well, the way how you can, like, if you go to supermarkets, like everything in supermarket has a barcode, yes, and then there is a reader. So the reader, if you just scan the barcode, the computer system understands what is it in your hand. We can use something very similar for those small pieces of DNA. We can attach to them a barcode, and barcode actually is also a DNA. It's a DNA which has a certain nucleotide code. It's very convenient. DNA can be used for carrying a barcode because you just make a certain sequence of nucleotides, which is unique, and you know that this sequence, this is a barcode for this patient, for this human, for this person, and another is for another one. And then when you make shearing and you break the DNA from the sample into pieces, you mix them with those barcodes, small pieces, and also there are also small pieces of DNA handlers. We name them. They're used also during the technological process in order to literally to take that DNA molecule, because we use the affinity, DNA affinity according to complementary properties of nucleotides. So we connect those handles and we connect the barcode to each piece. And after that, for each sample, meaning each patient, we have them. After that, we can mix them and process them together. That allows much cheaper price of processing. So, and besides that, of course, much cheaper price now is also because of advancements in software. So, but we'll look later, how do we use software? Now, what do we have now? We have this piece, fragment of DNA, which is like several hundred nucleotides long. We have those handles, and the process of sequencing, it used actually to take like 40 hours. Now I believe it's even faster. And what we do now? We add some blockers on ends of those studied DNA molecules. You see on the picture number 1. We add blockers in order to protect them so that they're not damaged, so that they don't connect to each other, etc. And after that, we should prepare the library. What is library? Okay, so every genetic analysis which we do, of course, we could read all DNA of a person, but usually it is not needed. First of all, if you remember, most of DNA of human is still black matter for us. We don't know what is it. We don't know how it works. So for research, yes, probably, but for medical purposes, we don't need to sequence it. We need to sequence probably only the exons. The exons are the part of genes which code the protein. That's exactly what we pretty much know how it works. And it is 1.5% roughly only. Besides that, in many cases, we don't need even the full exome. Sequencing of full exome is, of course, much more expensive than sequencing several genes only. So probably we want to sequence only 100 genes, 200 genes, which are meaningful for this specific patient, for his clinical picture, for making analysis. That's what doctor needs. So we can separate only certain pieces of DNA and remove everything else. And in most cases, it's removing more than 99% and sequencing less than 1% in many cases. What we can use? We have those pieces after shearing, and we can use, this technology is actually named fishing. It really looks very like, because first of all, we use baits, and the baits are some, again, pieces of DNA which have some small particle which we will use later to catch it. So that's small pieces of DNA will attach they will encode the portion according to the reference genome, which we know is in the piece of DNA which we need. So we connect them, and it is seen on number 2, on the picture number 2, those hybridized library fragments, which are, after we connect to them, we mix those baits, and the baits connect exactly to the pieces which we need, and others, they do not. And after that, we use special magnetic beads. And those magnetic beads you see on picture number 3, those roundish, ugly thing, big thing, that is a magnetic bead, which has an affinity to the bait. So those magnetic beads, they will connect to the bait. And after that, voila, you have the pieces of DNA which we need. They are connected to magnetic beads. Others are not connected to magnetic beads. What we can do? Add several magnets to catch them and wash everything away. So after that, everything what we don't need is washed away. What we do need is on our magnets, and we can start working with it. And what we do, we literally kind of put it into those small chips which use the sequencing technology. So we have those chips, we have the sequencing process in those chips, and from each sequencing process comes that sequence of several hundred nucleotides, which is read. After that, well, here's the problem. When we were doing the shearing, we broke DNA into small pieces. Now we need to connect them all together, right? We broke it in pieces because, as I told, we cannot read it from beginning to the end. There's lots of noise, chemical, physical, every kind of noise. It will be too many errors. So now those small pieces we have read. And here comes to our help the fact that the mutations or variations of DNA, they're not so numerous. Most of DNA code is the same, and we have the reference genome. It helps a lot because the reference genome is kind of how the standard genome looks like, and every person has several millions differences, places of difference. So if we have those pieces, and those pieces also, they mostly repeat what is in reference genome. And there are several mutations. Okay, so we can use, and here we use a software. So this is a software process which has the matrix of reference, that has those small files, pieces, and understand what matches where. And we can reconstruct how those pieces are standing in the order, connect, overlapped often, etc. And we can reconstruct the whole DNA, what we sequenced. And after that, we really also don't need the whole sequence. We need only the places where we have differences. So after we reconstructed, we name this process alignment. So we align all those pieces on the reference, and we can just extract again, it is done by software systems nowadays, and we can just extract those places which are the differences as records and put them in a file. And that will be literally the difference of this patient from reference genome. We know reference genome exactly, each letter. If we know difference, we know what is the genome of this person. You see the example of such record, for example, on this slide, which literally, you can read it, right? So the very first thing is a 6. It is a 6th chromosome. Okay, in 6 chromosomes, the second is the coordinate in nucleotides. What is that? It's 11 million, some thousand, something. That is a number, the offset of exact nucleotide about which this record is. And after that, you see, in the reference, it is C, but here it is T. So we have thymine instead of cytosine in this place on 6th chromosome. And everything else is pretty much kind of additional information about the precision, how we get the data, how reliable it is, etc., etc. So there are lots of additional data, of course, which are just part of technology. But that's pretty much the idea. So we get those files. Those files are very big. Those files, if you sequence the whole genome, it will be like several hundreds megabytes. It can be gigabyte of data or more. But that's pretty much the process which allowed us to reduce the cost of sequencing so much. So what we're doing after that? We have software which allows you to look literally gene by gene, nucleotide by nucleotide, the DNA. For example, over here, I show it on slide, but if you want, that software is in public domain. You can go ahead, download it, and play with it. On the very bottom of this slide, you see a link to the software. You see the link of international project which has genomes of many people. There is a project named 1000 Genome. You can easily research it online, and you can find many thousand full genomes of people sequenced available. You can get the software. And over here, what I show, you see is chromosome number 5. It's human genome. If you look on the top, it's written human genome, chromosome number 5. The coordinates after that is given 54 million something. That's the place in chromosome. And beneath it, you see the grayish stick. That is actually the chromosome, how it is shown here. Introns and exons and genes. All information is here. And the reference genome, and there are 3 samples of 3 different people. You see them in the very center, and you see that these are certain genes and how the places are shown, which are different from between this sample and the reference genome. You can zoom in to individual nucleotides, and you see right now I'm zoomed in certain genes. Actually, there is a pop-up window where you see even more information, which gene, which position, etc. And you can see that it is written that reference is cytosine and alternate is thymine. Yes, in this sample, the alternate nucleotide is thymine instead of reference. And you see on the bottom, the sequence, that is the nucleotide sequence. So it's very convenient for fun. Yes, but if we want something useful out of it, we need to understand which genes, which variants are these really, and what we can learn from those variants. I mean, what does it mean? Here we come to the next point, the point of interpretation. Interpretation of DNA sequencing or genetic analysis is it's very big discipline also, which also nowadays benefits a lot from computer science, because really there's a lot of data, a lot of variation. It cannot be done without very advanced cloud computing, which is modern technology used for it. So besides that, well, before we go to interpretation, what about RNA? Yes, the DNA is pretty much a mosaic of exons and introns. And we know that when RNA is synthesized, it goes through splicing process, and introns are removed and only exons are left, right? So you see how, and actually that's how it is. Introns are much bigger than exons. So RNA, which is used for synthesizing the protein, is much, much smaller than the original gene in DNA, because introns are really like more than 90% of the gene often. So we often need to make RNA sequencing also. Why? Because quite often the splicing process of removing introns does not go properly. And sometimes the variation which is in intron, and we would think that it does not affect the protein at all, but because splicing goes differently, this variation may get into your messenger RNA and will affect the synthesis of protein. Or sometimes there is some variation in exon, and because splicing went differently, it was removed in post-transcript processing, and it didn't affect the protein. So sometimes for some conditions, we need RNA sequencing also. Here comes some difficulties because unlike DNA, RNA is very unstable. It needs a special handling, it needs a low temperature, it needs to be processed very quickly. RNA is unstable, it breaks quickly. DNA is quite stable. That's why in our cells also, DNA is used for long-term storage, right? Chromosomes. And RNA is used for very short-term storage. Just transcription, translation, synthesizing protein, and after that RNA is destroyed. So there are more technological difficulties with RNA sequencing, but it is also done and it is also useful. Now we come to the interpretation point, right? So we talked about the sequencing, the alignment, and variant calling. Variant calling, actually, that's also where the reference genome helps us, because we understand that every human's genome is pretty much like reference genome with just some different small places of differences, just several millions. For billions and for the size of genome being several millions places is less than 1%, as you know, less than 0.1% even. So after that, we need to understand the variants of genes, what they are. Are they benign or are they pathogen? Or are they unclear? We don't know what are these. So we need to annotate the variants, we need to annotate the genes, because we need to understand what is this gene, what does it do? Is it related even to the clinical conditions which we observe? And we need to prepare the clinical report. Over here, computer science advancements are critical. The big data, literature mining, using natural language processing, in silico experiments and protein folding analysis, which is pretty much also in silico experiments. That's the point which I'm about to touch now. Well, first of all, the big data, right? So of course it is big data, and of course there is many combinations, right? We have like 20 to 25,000 genes in humans. How many combinations is this? It's a lot of combinations. Like if you take the factorial, you cannot take factorial of 25,000, right? It's an astronomical number. So we cannot analyze it with that. It's intractable by enumeration. That's how we tell it. We cannot analyze it really, but we can make some approximations. And here some machine learning techniques come into the game. In order to make the machine learning and our models to work better on that data, we need more data, right? We need more data for training our models to understand how those variants will work, to good or to bad, or they do not change anything. So over here, actually, commercial companies probably are in better positions than academic institutions, because how many results of genetic tests can academic project have? And commercial companies have millions of them. So using a company which did sequence, like the company where I work, we did sequence like many millions of people. We have lots of data in our database, which allows to process it and to build better models to understand better variants of those genes and how it works. On another side, we always do literature mining in order to understand what are findings and publications about specific genes and specific variants of genes. Biomedical literature, all researchers, even not only academic, commercial companies also, they publish information, the results of researchers. Those results of researchers may contain information about certain gene and even certain changes in a certain gene, and how does it change the clinical picture, right? So how many articles available on medical resources online? You can pull all that data and analyze that using natural language processing, which is one of the branches of computer science. You can analyze it and extract data from those medical records and medical texts, understanding more about variants and using that to make interpretation of those variants. And besides that, so that is literature mining. And besides that, we use an in silico experiment. Well, pretty new terminology probably, because we always spoke about in vitro, in vitae, and in vitro experiments, right? In vitae are in living, meaning experiments with living organisms or culture, and in vitae, in vitro, it is in glass. That's what it means in Latin language. In glass, meaning it's in laboratory with those vessels, and we mix certain reagents, and that's how we analyze. Well, experiments in silico, it's experiment which is modeling in computer. So it is an experiment which can be run in software, by software. We can model certain things. And let us think about it. Well, let us think about physics, right? In physics, if you know very well the Newtonian laws of mechanics, you don't have any problems with understanding how balls on pool table or billiard table will move, or how the soccer ball will roll across the soccer field. You can predict it with very high precision, right? Just based on several formulas of physics, of mechanics. Because we know those laws very well, and because there's not so many factors, and with biology, it's much more complicated. As I mentioned, there are 25,000 genes. They all affect each other. There's so much about epigenetics, the methylation of DNA, etc. There's so many processes which regulate the transcription and translation. There is a lot of data to take into account. So that's why we cannot really model it by just several laws just like with mechanics. That's why we need machine learning. In physics, you don't need a neural network to model how the soccer ball will roll across the field. In biology, we need neural networks, we need machine learning, we need to understand it by approximation, etc. But many things also can be better understood by running experiments, and those experiments can be made in software by modeling. If we understand certain laws, we can run it, encode those laws into the software, and run it as an experiment. And one of such advancement was done in understanding protein folding, which is critically important for interpretation of genetic tests. Look a moment. This is a very famous picture, right? Or you probably saw something like that. It is a genetic code. How 3 nucleotides in the triplet of nucleotides, codon, encode the amino acid. And you see over here, and we of course know that change of nucleotide sometimes does not change the resulting amino acid, because there is some redundancy. For sometimes there's several codons which code the same amino acid. So the third nucleotide is important. So quite often you can change it and it will be the same amino acid. So it doesn't change anything. Okay, over here we understand. So reading the nucleotide sequence and knowing this genetic code, we can understand, okay, this mutation, the nucleotide changes, but amino acid will be the same. So no difference. But there is more to it, because the protein consists of multiple amino acids. And after protein is synthesized as a result of translation, the protein goes through folding process, when that long chain is folded into big globe, and the geometry of that globe is important for the function of protein, and the surface, probably the amino acids which are on the surface of the globe is important for function of the protein. But if the amino acid is somewhere inside, if it stayed inside the globe because of the folding, probably it did not change the function of protein. So quite often you can change the amino acid in the protein without changing the function of protein at all, or changing it very insignificantly. But in order to prognose it, you need certain experiment. You need to understand how will that protein fold. And for a long time, we do have technology to understand the geometry of protein, but how to understand from nucleotide sequence to amino acid sequence, the polypeptide sequence original, but then how does it fold? How the geometry of that protein happens, the 3D geometry. For a long time, we didn't have the tools. Finally, it has. You see, it is rotating here. I specifically show the animation because it is 3D shape, right? And we now have a software named AlphaFold, and there is AlphaFold 2, actually a newer version, which makes prediction of the folding of protein with very good precision. On those animated 3D pictures, you see two proteins, and experimental result of how the protein folding goes is shown in green color, and computational prediction is in blue color. And you see that they're almost precisely aligned to each other. So we can use this technology for more understanding and interpretation of the result of our genetic tests. Yes, because if there is some mutation and we understand the amino acid changed in the gene, we can understand how geometry of the protein changed and understand then how the function of the protein changed. And if we understand how the function of the protein changed, we can understand the biological or physiological, biochemical consequences of that change. So that is very important. We were speaking a lot about genetic diseases, right? But there is one more branch in medicine where mutations of DNA are extremely important. And that branch is oncology. Cancers, tumors are pretty much factories of mutations. Their DNA is quite unstable. They mutate on scale. And understanding that helps us to understand cancer. Helps us to understand cancer and also individually tailor a certain treatment strategy for that oncological process. Well, speaking about that, oncology and genetics, probably we usually mention proto-oncogenes and tumor suppressor genes. Those are two things, mutation in which can cause cancer. First, we discovered proto-oncogene. First, we discovered tumor suppressor genes, and we discovered the specific gene BRCA1, which was named because of breast cancer. And actually, yes, there is BRCA2 also. And we discovered that a mutation in this gene causes cancer with quite high probability. And then we understood why. Because this gene is a tumor suppressor. It is critical for the processes of suppressing tumor growth and making the tumor more vulnerable for our immune response eventually if we suppress its growth. So mutation in this gene makes the proteins, or the result of the gene work, compromised, and that allows the tumor to grow. That's how mutations in tumor suppressor genes can cause cancer. And on the other side, we have proto-oncogenes. These are the genes which are usually responsible for something else, something related to differentiation and function of, or some control of cell proliferation. And if it is mutated, that goes out of control and cancer happens. We now know already more than 70 genes associated with higher probability of oncology, which are germline, which is kind of a genetic disease, you can tell, or something inherited. And we know much more than 300 of somatic mutations, which are very characteristic to certain kinds of cancers. And what we found also that different kinds of cancers, depending not only on the histological profile, but also on the genetic profile, they can respond differently to different treatments. That's how we developed already more than 100 precision medicines. And precision medicine is a medicine which is like very well targeted to certain kind of cancer, depending on which gene mutated. You see several examples here on this slide. So that is one of methods of diagnostics, actually, which we use. It's a CRISPR diagnostics in treating cancer. CRISPR diagnostics is not really connected to the precision medicine, but it's often used. And what does it do? CRISPR is a technology which was discovered also relatively recently. And it was discovered in certain bacteria, and CRISPR is used by bacteria to combat certain viruses which affect those bacteria. But CRISPR technology was taken by scientists to actually a very useful instrument. There is a protein which acts like a scissors for DNA molecule. And what is good about that protein, that you can prime this protein with a certain piece of polynucleotide, and that piece of polynucleotide writes certain code, and it will allow that scissors to attach to DNA in specific place, according again to affinity of polynucleotides to nucleotide sequence in the DNA. And it will cut in exact precise place. So we can use this to make genetic engineering to change DNA, because we can cut DNA in certain places, and then we can use polymerase to include additional nucleotides. But we can use it also for reading DNA, for diagnostics, and it becomes quite convenient instrument for DNA analysis. So we can analyze cancer using this or some other types of genetic profiling of tumor, understand which mutation, and depending on different mutations, we can assign different types of treatment for the person. That's one more benefits. By the way, it's very similar to that. Recently, we started to consider an experiment with sequencing the bacteria in case of certain infections in order to quickly understand the resistance of bacteria to different proteins. You know that resistance of bacteria to proteins can be used microbiologically by taking those bacteria and starting to grow them on a substrate with those antibiotics. Sorry, I talk about resistance to antibiotics. So it can be used by bacteriological analysis, resistance to antibiotics. But we understand also that resistance to antibiotics in bacteria is because of certain genetic variations. And we can do sequencing much faster sometimes than we can do bacteriological analysis. That's one more application. And there is another application, and that is named liquid biopsy. What does it do? What is it? Well, you see on this chart interesting information about the mortality of cancer and how much it is connected to metastatic cancer. Metastatic cancer is like less than 20%, I believe it's here, like 17% of cases. But 55% of all cancer deaths is because of metastasis. Why does it happen? Because it gets unnoticed and we don't catch it early. A person with a tumor goes through oncology, goes through some other treatments, and then returns his normal life. And several months later or several years later, a metastasis of the tumor may start growing, and it may affect vital organs much faster than we diagnose, than we notice that. So it is vitally important to early diagnose the metastasis. Of course, there are instruments how to diagnose metastasis. We have ultrasonic, we have X-ray, we have radio diagnostics, etc. It's quite complicated. It disrupts the style of life. It also can be quite expensive. Often it happens that patients are not able to do it frequently enough for some reasons. But here, genetic analysis can come to help. Why and how? Tumors leak DNA. Well, all cells leak a little bit of DNA to blood, but tumors leaks more of DNA to blood because of how biochemical processes go in tumors. DNA in blood, if it is tumor DNA in blood, well, what if we really take the blood from patient and then analyze it? So what we do literally, here are the 3 steps. A person has tumor. We can take the tumor, we can make DNA sequencing of that tumor, and we kind of understand the genetic variations of that tumor. We understand the specific fingerprint of that tumor, how this DNA sequence look for this tumor. And after that, after the surgery, months later, years later, we can just take blood sample from the person, and we can run DNA sequencing and see if we find that tumor-specific fingerprint in blood. And if we find it, we understand that there is a metastasis growing somewhere. Okay, you should go for more specific laboratory analysis. But this liquid biopsy is quite convenient, minimally invasive way because it's just sample of blood. So it is convenient. It saves the lives of people. Well, we covered a lot now in presentation, but of course there's a lot more, and there's a lot more still unknown. We talked that exons, the portions of our genes which code proteins, they're less than 2%. You see here the gray circle, it's pretty much the whole DNA, and the orange circle in the middle, that is the exome. So that's how much the gray around, that's how much we still don't know, don't understand how it works. And even in that orange, which is a full exome, not all genes we understand. There are 20,000 genes, we probably understand like 5,000 or even less than 5,000 of those genes, what clinical results they have. So that is a green area which you see, yes? So probably we often do the full exome analysis. Quite often we do the clinical exome analysis, which is smaller, it's just 5,000 genes. But most of cases, for clinical purposes, we can make analysis of just several hundred genes. That's what we name panel or assay, because that's what is named the MGPS here. Usually it's several hundred genes. That's a very small piece. So you see this yellow dot, that's what we do usually. Well, on one side, it allows us to make it much cheaper. On another side, you see how much unknown is around it. Besides all this unknown, non-coding, or part of DNA which is not coding proteins, which is unknown, there is a much more, because the proteins, even after transcription and translation, proteins do go through certain post-translational modification. We talked about folding. Folding can go in different way even, but there are also some chemical changes which may happen to proteins, and there's phosphorylation, glycylation, methylation, acetylation, etc., ubiquitination. So those changes may change the protein which was synthesized on messenger RNA. That's why it is considered that the proteome, and proteome is a composition of all proteins which are available in human body. So proteome can be at least 3 times more complex than the genome, probably even more complex, because proteins, yeah, they differ not only from cells to cells, they differ from time to time, and proteins can change. So more more research is needed, of course, to understand human software. Human software is not only the DNA. It looks like there's a lot of human software. That's what I called it. Yes, the code or like how organism functions. It's not only defined by DNA. A lot is defined by how proteins interact and change. Proteomics is a new branch, relatively. It's really like we just scratch the surface. We don't know many things. And beyond that, you see another thing here. It is regarding microbiome. What is microbiome? Well, humans have more bacteria than our own cells. And if there are more bacteria than our own cells just several times, but if we speak about genes, there are more than 100 times more bacterial genes in our body than our own genes. And those bacteria and their genes do affect our health. It is already well understood that bacterium does affect our immune system. It does lead to allergies. It does lead to autoimmune diseases. Actually, one of the books which I prepare right now is on autoimmune diseases and repairing immune systems through changes of microbiome, which people can do by changing lifestyle, changing nutrition, etc. But what is also interesting is that this is a genes which are in our body, which also affect our health. But first, they can change. They're quite volatile. Unlike your own genes, you cannot change your own genes. You inherited it, but you can change your bacteria, which means you can change genes, many genes in your body, which are not your own, but your microbiome. And also, people do have a lot of differences in microbiome. If you see this chart on the very bottom, if our own genome is like 99.9% identical to each other, our microbiome genome is just 80 to 90% identical. There are more than 10% differences. There are so many more things to study and understand. On this point, probably, I will finish the presentation and we will pass to questions and answers. Thank you. We finished the presentation. We can pass to questions and answers.
Thank you. Thank you, sir. Now the floor is open for question and answer. I'm requesting the participants to type the questions in the chat box, or you may ask directly. Unmute yourself and please ask.
You can also type it in chat if it's convenient. I'm reading the chat, so.
I'm requesting the participants. If you have any question, please type them in the chat box.
Okay. If there are no questions, then, well, thank you. Okay.
Sir, I have a small question.
Sure.
In the later presentation, you have mentioned about the diagnosis of in cancer, the treatment of that coding, and you have mentioned it. What is that latest advancement, sir? Is that in process? Is it going on to giving the treatment for the cancer?
Well, genetics processes are definitely used for diagnostics treatment is a different branch, but I mentioned several. So which one do you use? A liquid biopsy? Liquid biopsy is used for early diagnostics of metastasis. And tumor profiling is used for understanding the genetic composition of cancer, because as you know, like if certain genes are mutated, the proteins inside the cancer will change, the biochemistry inside the cancer will change. If we understand that, we can target with our medication specifically certain proteins or specifically certain biochemical processes inside the cancer. And right now there are already like more than 100 different medications which are very precise for certain tumors of certain genetic composition. Previously, it wasn't possible because we knew only histological picture. We knew what we saw in microscope. Now we can understand what is happening inside the cell. And of course, there is more research in that.
Yes, thank you, sir. And also, we are really happy and we are enlightened with your presentation regarding the decoding and software advancement in the analysis, genetic analysis, and finding the Down syndrome, those things and all we really learned. And thank you so much, sir. And now I request. Yeah, now I request Dr. N. Vimala Devi, Head of the Department of English, to propose vote of thanks.
Unmute చేసుకోండి మేడం. మేడం అన్మ్యూట్ చేసుకోండి. మొబైల్ అన్మ్యూట్ చేసుకోండి. మైక్ సింబల్ దగ్గర రెడ్గా ఉంటుంది చూడగా. ఓకే.
I can't hear.
Yeah, am I audible? Yeah. A warm and graceful morning to one and all. Honorable dignitaries, distinguished guests, respected speaker of today's session, Dr. Anna Gulio, and dear participants. It's my privilege to propose a vote of thanks and acknowledge the contribution of those who work really hard to make this international webinar possible. I, Dr. N. Vimala Devi, Head of the Department of English, on behalf of J&J College for Women and the entire fraternity of the institution, extend my sincere thanks to the Almighty for making today's session a grand success. I take this golden opportunity to thank our guest speaker, Dr. Anna Gulio, lead software engineer and architect with a medical degree in vitro scalable genetics and oncology, Azerbaijan Medical University, Dallas, who spared time from his busy schedule to grace this webinar with his extremely relevant address on decoding human software. Today, we had an opportunity to hear your thoughts, and we have got a lot of information on oncology and genetics. You truly made this day enlightening for us. Sir, you gave deep insight into the human genome, genetic defects, genetic analysis, genetic code, and Down syndrome with a statistical significance, and revealed some interesting facts. Dr. Anna Gulio rightly stressed and talked about man's innate capacity to adapt and invent new strategies to overcome challenges, which is very much required in the present times of pandemic. So, we have no words to offer gratitude for your valuable presence and wonderful presentation. Thank you so much, sir.
Thank you.
Thank you, sir. I would further extend the hearty thanks to Dr. Sister Teresa Gade, corresponding, for always encouraging us and providing opportunities to organize such events. My sincere gratitude goes to Dr. Sister Shaini, our principal, for being a constant motivator and guiding force in all our endeavors to excel in the field of education. My deep sense of thanks and appreciation to the organizing committee, Mrs. M. Aruna, convener, and Dr. S. Uma Maheshwari, organizing secretary of the webinar, for taking topics that really matter into this age and time. I would especially thank to all the participants who choose to be live with us and attend the webinar with great enthusiasm and made it successful. Last but surely not the least, I thank Mr. Murali for all the technical support for helping us webcast our webinars. Once again, a big thank you to each one of you wholeheartedly for glorifying this international webinar with your gracious presence. Have a wonderful day ahead. Thank you. Thank you, one and all.
Thank you.
Sir. Thank you so much. Thank you, sir.
So this concludes the webinar. Thank you all for attending this webinar. We hope you have learned and enjoyed today's session. We look forward to the same cooperation in the following days as well. Today evening, we will send new link for tomorrow's session. Thank you.